Okay, here we go. Here's the physiology, the how of what's happening during an actual muscle contraction. I've broken this down into 4 phases, and each one of these, hint, could be a short response question on the exam. I love this stuff. So excitation, this is the process of getting the muscle excited. So it's going to receive an action potential from the nerve and start an action potential in the muscle. So if you follow along with the book, it has the same numbering scheme. You can use these steps to write a short response essay. So the very first thing that's going to happen is the arrival of the signal. The action potential, so that's the signal, is going to come down the nerve to the axon terminal. Once that action potential makes it to the end of this neuron, it's going to stimulate the release of the synaptic vesicles, and you can see all the little green acetylcholines being released into the synaptic cleft. All right, now the picture is going to zoom in here on these receptors. So here's a single receptor. Here's my sarcolemma. These acetylcholines, my little green ligands, are going to bind to my receptor. That is going to open an ion gate. So this gate is a facilitated diffusion, and it's going to open once these bind. So it's kind of like a lock and key. So these are the key, and they open the door. If you remember, we had a lot of sodium on the outside and a lot of potassium on the inside. So now these are going to go down their respective concentration gradients. So this is going to be diffusion. And it's going to allow sodium to come in and potassium to go out. When sodium comes into the sarcolemma, it's going to create an action potential. So this is going to change the voltage across this membrane from -70, which was resting, to some positive number, because we're moving positive in and the fewer positives out. And this is going to start the action potential. So excitation is going to end here because the muscle is now excited and it's ready to go ahead and start telling itself we're going to contract. So my second phase here is excitation, contraction, coupling. So what does it mean to be a couple, right? Coupling means there's 2 people or 2 things together. Well, here are my 2 things. My coupling is going to put the muscle cell being excited with the actual contraction. So there are a few steps between the muscle cell being excited way out here and the actual muscle in here physically contracting. So all of these things are going to connect those 2 together. So we have an action potential. The action potential is going to travel down the T tubule. This is how it gets to the middle of the muscle cell. Once that action potential travels down this T tubule, it tells those terminal cistae, those terminal cisterns right here. Remember, this is the sarcoplasmic reticulum, and these are going to release the calcium. So these little yellow or gray dots here, those are the calcium being released into the actual muscle cell. So you can see here's a sarcomere, here's a sarcomere, here's a sarcomere. And they're getting a whole bunch of calcium flooded throughout all of the sarcomeres. So now we're going to zoom in. We're zooming in here on a few of the myofilaments. Here's actin, troponin, and tropomyosin. The calcium will bind to troponin. And when calcium binds to troponin, it changes its shape. And when it changes its shape, it changes its function. So when calcium binds to troponin, it's going to move the tropomyosin. So normally tropomyosin is covering these little active sites. You couldn't see those in the previous picture. But once the calcium binds to troponin, tropomyosin moves out of the way, and these little active sites become visible. Those are the myosin binding sites. Myosin binds here. So when they're covered, even if I have a little myosin head, it can't actually connect, right? Now that this is open, the myosin has the ability to actually grab onto the actin, which is how we're going to start with our contraction. So we had to get the excitation signal all the way down into the cell, release the calcium binds to troponin, the troponin and calcium moves tropomyosin out of the way. Here's the next phase. I really like this one. So this is the contraction. During contraction, the muscle fiber, remember this is the entire cell, is going to shorten. The individual filaments don't actually change size. So the sliding filament theory is that the myosin and the actin just slide past each other. They're not actually going to change how big they are. And this is going to work in a cycle until the contraction is over. So we're going to start here. We've got calcium bound to troponin. Tropomyosin has moved out of the way to make these openings available. So an ATP will be hydrolyzed, meaning that the ATP is going to be broken down into ADP plus inorganic phosphate. When that happens, the myosin head cocks back. It's getting ready. Once the myosin head is in the right position, it can actually connect to the actin binding site. So this is going to form a cross bridge. So a myosin-actin cross bridge is where these actually connect together. Think about like putting Legos together. The ADP and the phosphate are released. This is going to allow the myosin head to return to its original position. And this is called the power stroke. So this actin moves from where it was here to here. So it's moved this much distance. And so this is how the myosin is going to pull the actin in this direction. And it does this over and over again. So once it's pulled at a short distance, we're going to bind a new ATP to the myosin head, and then hydrolyze that ATP to cock the head back again and pull again. So a contraction of a muscle is actually hundreds of thousands of tiny myosin heads being cocked back, forming a cross bridge, power stroke to pull the actin, release your ADP, get a new ATP. Get it cocked back, form a cross bridge, pull the actin, get a new ATP. So this cycle is going to continue as long as this calcium is here. As long as the actin fibers are eligible to bind with the myosin, and as long as we have ATP available. To power this, this is going to continue to turn. But you don't want your muscle to contract forever. That's called tetanus, and that's not a good day. So at some point, at the end of a contraction, we have to relax. So relaxation starts with the nerve. Contraction and excitation started at the nerve. It ends at the nerve. So eventually, the signal will stop coming down the nerve cell. When the cessation of the signal stops, it no longer releases acetylcholine, so these are not being released anymore. We already had some acetylcholine out here. Some of it gets broken down. So this is acetylcholine esterase, which you don't need to remember that name, but we've got an enzyme here that's going to get rid of some acetylcholine. We also have to deal with concentration gradient. So when we're not pumping a lot in here, the ones that were on here are going to unbind. So in some ways, the acetylcholine that was here is going to be removed from the receptors on the muscle cell. Once those receptors are removed, the action potential stops, and calcium is going to go back into my sarcoplasmic reticulum. So we're going to reabsorb the calcium back into my blue sarcoplasmic reticulum here. When we do that, we pull the calcium away from troponin. So we're removing calcium from troponin, and when it does that, it returns to its original shape. And now tropomyosin is covering those actin sites, And even though we've got ATP here, myosin cannot form the cross bridge. And so contraction cannot occur. So the contraction at this point is over and the muscle fiber will relax. So take a moment, more than a moment, and reflect about the steps during contraction. Remember, contraction was the third slide that I just went through.